Good morning, everyone, and welcome to this first lecture. As was introduced, we talk today about modeling neuron-astrocyte interactions, and this is not a conventional topic in this summer school, but we have the recent years we have taken it as one kind of short lecture to teach you about these aspects of the brain. So, as you learned during my introductory lecture, in addition to this about 100 billion neurons, our brain has almost an equal amount of glial cells. And what are these glial cells? So they are non-neuronal cells, both in the central and peripheral nervous system, and they contribute to various functions, both in the developing and also in the adult brain. And we will look at these a bit later. What is many times not known is that during evolution, when neurons, when they were joined together, there was an appearance of these non-neuronal supportive cells called sometimes also neuroglia. And this happened when the nervous system evolved from the diffuse to centralized form. So it has been like the process during development that we kind of ended up having so many glial cells, almost the equal number to neurons. And what we know today is that the glial complexity and number increases with the overall complexity of the nervous system. So this is kind of general fact that you can take from the glial field. What is also not known that glial cells in general, I think it was kind of like the term glial cells, we didn't know that astrocytes existed at that time, but glial cells in general. So they were discovered by German pathologist Rudolf Virchow already in 1856, so very early on. Before we move to astrocytes, we look at a little bit like what types of glial cells there are. So the first thing to know is that there are these Schwann cells and oligodendrocytes that form the myelin in peripheral and central nervous system. Then there are important cells called microglia that take care of the maintenance of the brain. Then there are astrocytes that reach both neurons and the vascular system. And then there are also other types of glial cells like these ependymal cells, so they control the cerebrospinal fluid, radial glia that guides the development of neurons, and they become Müller cells and retina, Müller cells in retina and Bergman glia in cerebellum. And there are also NG2 cells that can become either oligodendrocytes or gray matter astrocytes. And maybe there is also one other type, but I have forgotten it today. You can ask Laura. Today I'm going to talk briefly about astrocyte biology, then computational modeling of astrocyte functions and activity and functions, and then I take 3 examples very briefly, and then at the end we take-home messages. And let's hope that I can leave you enough questions, time for questions at the end. So, already more than 100 years ago, Ramon Cajal showed astrocyte-neuron interactions in the brain. So in this illustration, you see these dark brown cells, neuroglia, that typically have small soma, thick processes and fine distal processes around pyramidal cells in this case. So for example, there is this in pink, this big astrocyte that is kind of close to the pyramidal cell, and then you also have twin astrocytes forming a nest around another pyramidal cell. So this was shown already 100 years ago, but we have mostly neglected these cells because we thought that they are only there as a glue, supportive cells. So let's jump to this day, and today we know that human astrocytes are far more complex and larger than their rodent and primate counterparts. So here you have a gray matter protoplasmic astrocyte in human and mouse, and then the same in white matter, there is typically this kind of fibrous astrocyte, and this is for human and this is for mouse. So when we compare the morphology of mammalian neurons and astrocytes, so we see that for mouse, monkey, and human astrocytes, there is an increase in size and complexity of pyramidal cells, for example, between mouse and human. But what we don't normally think is that when we compare the cortical astrocytes from mouse and to human, we see large, I would even say a huge increase in size and complexity of these astrocytes when we move from mouse to human. So this is something that is not discussed so much in the talks and literature and books. The reason for this is that it's only very recently, actually, that we've been able to visualize these cells and then also reconstruct them. So it has happened very, very recently. Astrocytes also cover blood vessels and neuronal dendrites. So in this illustration or reconstruction, which is courtesy of Corrado Calli, now in University of Turin, and at the time when these reconstructions were done in Pierre Magistretti lab at KAUST. So in this reconstruction, you see astrocyte as a green element, blood vessels as red, and neuronal dendrite as a gray elements, and you see this huge coverage of a cortical astrocyte in the brain. So the number of human glial cells vary between brain regions. So this we already know, but we don't know the exact numbers. I have taken here some estimates. So for example, in the cerebral cortex, we think that it's about 4 astrocytes to 1 neuron. In cerebellum, perhaps 0.23 astrocytes to 1 neuron. And in the rest of the brain, very surprisingly, at least to me, it's 12 astrocytes to 1 neuron. And of course, here we have to remember that it's all glial cells that I listed, not only astrocytes that we are now mostly discussing in this talk. But what I wanted to kind of point out here, it's from this part health, where also some of these, or most of these numbers are, but there is also other supportive literature. So it's stated that more attention must be paid to quantitative studies of neuroglia and also nerve cells, as opinions are often conflicting and frequently based on faulty techniques. So we really need to improve in this field as well. We also know nowadays, since 2020, that cortical astrocyte layering is distinct from neurons. So here is the cortical neuronal layering, and you see the layering at P14 animal for astrocytes. So they have these 3 layers, superficial, mid, and deep layer, that then merge into 2 in P56 animal, basically the adult animal. And this is done based on single cell in situ transcriptomics. The white matter layer 6 astrocytes, they are maintained into adulthood. Then about the roles of astrocytes. So they are indeed very important and they have important roles in many brain functions. So first of all, and basically all these roles that I list here are known for many, many years already, like 10, 20, 30 years or even more. So we know that pre- and postnatal development, they initiate neurogenesis, they structure the brain, and very importantly, they mature and prune synapses, and this we will look at a bit later on as well. In energy metabolism, they support blood-brain barrier functions, they control glucose and provide nutrients. And very, very importantly, and this is also, I think, known for 20, 30 years, they do these homeostasis and survival functions. They, for example, maintain extracellular ionic balance, potassium and other ions, and they take up neurotransmitters. And these, we know that they influence the way brain functions. But how they do it mechanistically, like at the level of neuronal activity, neuronal synchronization, brain states, and how they change brain states, we don't fully understand yet. And there we really need the models that I'm going to soon present. Astrocytes are also implicated in many, many neurological neuropsychiatric diseases as listed here. But what is a bit more controversial, although it's known that they mature and prune synapses, so we have only recently started to get like more evidence about the role of astrocytes in plasticity, learning, memory, and cognition. For example, like how they control synaptic transmission, synaptic plasticity, and so on, and in which conditions and in which developmental phases. So this may differ. So they may not do the same thing depending on the condition or on the developmental stage. So over the past 10 years, as I said, more and more evidence about the role of astrocytes is accumulating. And basically, like this year or last year, every week there is a Nature or Science paper about role of astrocytes in the brain. And here I just list a few examples, like astrocytes modulate network activity, brain oscillations, state transitions, and sensory processing. You get some very high quality publications as an example. Astrocytes modulate cognition, behavior, sleep. Astrocytes modulate synaptic plasticity, learning, and memory. But here I have to say that there are controversies, which we don't have time to go, but I have written about those like in a little bit more layman manner in some of my publications, so you can look at it, the controversy there. So I conclude this very high-level presentation about astrocyte biology and physiology by stating that we really need additional studies to better understand how astrocytes regulate brain activity and functions in awake animals. So it's important to realize that many of those studies, they are either under sleep or a slight anesthesia. And many data also comes from in vitro or slides. So we really need studies in awake animals. Now we slow down the gears a little bit and go 30 years back in time and look at where the study of astrocyte physiology started. So when it was discovered that the astrocytic calcium activity is actually very important, and these discoveries were made in vitro. So there were many research groups at the time, very active research work done on isolated astrocytes and cultured astrocytes. And I was also at the time involved in some of these studies, in this case in Professor Kimmelberg's lab, late Kimmelberg's lab. And we frequently saw these kind of calcium signals recorded from astrocytes. So in cultured astrocytes, you typically had this kind of peak, which kind of resembles almost an action potential, but it's not driven by sodium, it's driven by calcium. And then this kind of oscillations, depending on whether it's spontaneous activity or whether you stimulated the system with neuromodulator like 5-HT or so on. We also did already early on some modeling of these signals, like the same way you model the action potential in neurons, in kind of trying to understand what are the mechanistic causes of these signals, or what is mechanistically behind these signals. But we already at that point, we saw that it's very complex. Some other groups also followed like how the spontaneous calcium activity spreads in these kind of isolated cortical astrocytes with time when you stimulate the astrocyte. So nowadays we can, or several research groups on different continents, they measure astrocytic calcium activity in slices and also in vivo in this way. Here is a 2-photon time-lapse imaging of these calcium transients in cell soma, and then also in these branched processes. This is work from Min and Nevian in their paper in Nature Neuroscience, and we have used this data in our modeling as well. So, from this very brief introduction to astrocyte physiology and biology, I would like you to remember that the calcium activity in soma and process is a little bit same way as you consider the excitability in neurons. It's different between soma and these processes or end feed. So end feed is the process that kind of reaches to the vascular system. And these signals differ in frequency, kinetics, and spatial spread. And with this slide, I conclude my biology presentation and tell you that, like in the search of trying to understand the source of this calcium signal, we have ended up this kind of picture, and I think this is still incomplete. So there are listed those mechanisms that are verified, at least in vitro and in slice. So we still don't know in vivo, but most likely also in vivo. So you see it's very complex, these mechanisms like how astrocytes build up these calcium signals in the cell soma and then in these processes and end feed. And if you are interested in understanding more about this, you can look at these review papers where these things are explained. And now with the models, we are trying to first of all reconstruct the calcium signal and then further on to understand like how these different mechanisms influence, for example, like in this case, the signal transfer in synapses. So let's then move to computational modeling of astrocytic activity and functions. So I start stating that there are thousands of models for different types of neurons. They are very detailed. We have tuned the channel kinetics based on the recordings from the very same neuron. So at the same time, we found out this is a study done a couple of years ago, so now it's a bit more. I think it's now like 300 models, but at the time there were hundreds, like 100 models, now it's hundreds. We studied about 106 of these models, and we concluded that there are like single astrocyte models, astrocyte network models, single neuron astrocyte synapse models, and then neuron astrocyte network models. So all types of models have been built with relatively little knowledge about like how this system works, or with very little anatomical details. We ended up also looking at the mathematical models themselves and came up with this kind of complex figure. I tried to briefly explain it to you. So the same color here always indicates that the next model is basically based on the earlier model. So it's exactly mathematically, not even a single parameter value may vary. So basically all these models are either this type of called de Jong-Kaiser combined with Lee-Ringel approach, a little bit more phenomenological model to reconstruct the calcium signal, and then another more biophysical Hofer model. So there are like basically three different types of models here, and then these green ones are individual, slightly different models. So the situation is completely different than for neuron modeling, where we have like specialized models for each cell type in the brain. What type of functions these models typically describe? They try to, like I said, reproduce calcium dynamics, model homeostasis, vascular coupling, synchronization, information transfer in the brain, synaptic plasticity, hyperexcitability, for example, in epilepsy, and then look at the diseases. But with almost the same type of very generic model that actually stems from the data done in oocytes. If you are interested in learning more about these models, so we have this paper and then a book chapter, which is a more kind of educational. So what type of methods you typically use when modeling astrocytes? So if you are modeling this kind of, or using this kind of well-mixed system, you typically use ordinary differential equations, stochastic differential equations, or even chemical master equation. And this is the same as basically for neurons. But many also now are starting to do really kind of like detailed modeling using reaction diffusion and so on. But you learned yesterday in the presentation by Ausa Saudarkiene the type of model, like how you model this signaling pathway. So basically for astrocytes you typically use the same approach. So you have two substrates reacting and you make a product, and you use this kind of ordinary differential equations to describe this signaling that we see intracellularly in the astrocytes. And here is an example that typically because in astrocytes, the main source we know this is the endoplasmic reticulum. So this is the main source of calcium. So this is always modeled, and you typically model this leak channel, then this Cerca pump, and then this IP3 receptor using this approach. But there are also many other calcium sources in astrocytes, as you saw from the earlier presentation, and we are in a process of describing those as well, but we are lacking data. We have also analyzed these models, published, like what type of inputs they use to activate the calcium, what type of variables those models have, and what type of calcium fluxes they describe. And as you see, most of these models, they do describe exactly the same things, and also with the same mathematical equations. We have also addressed extensively model reproducibility because we were interested in using these models in our work, but we concluded that of those models that we have studied, only few models can be reproduced. You can see here that only these two models are really great, so we can reproduce all their results. And several publications, they lack details how to implement the model, and only few are available in databases. And what is a bit alarming to me that the same physiological phenomena, like I said, is explained with the very same models. And to me, this is a bit like interesting that it's not compared to the other models that describe the same physiological phenomena. So I'm a bit critical with the existing astrocyte models, and I want to kind of inform you about it. You can look at more from our paper in Frontiers in Neuroinformatics. So then we should move on to my examples, and I'm checking the time. So I will very briefly take you a tour through this very complex work that we did. It took 10 years, almost 10 years. And the main actual modeling, but it's more the controversy that exists there that prolonged the work. So this first example is about neuron-astrocyte interactions in synapses, and this is a collaboration with Tiina Manninen and Ausa Saudarkiene. So we learned at the beginning of the lecture that astrocytes have a fundamental role in activity-dependent synapse pruning. So they control structural synapse formation, functional synapse formation, and control of synapse elimination. And there also microglia is important, but we don't go into it at this point. So this is work from Allen and Eroglu and many others. So this is known already for decades. At the same time, there are now studies, for example, this Min and Nevi in 2012, where they showed that astrocytes take part in long-term plasticity in developing somatosensory cortex layer 4 to layer 2-3 synapse. So they were recording from pyramidal cells and also visualized cortical astrocytes. And like you saw, they were visualizing like how this calcium activates in the soma and processes and so on. And they use this pairing protocol that Ausa Saudarkiene explained yesterday to induce STDP. And what they found out that this STDP induction is dependent on endocannabinoid release from astrocytes, and this induces calcium signaling in astrocytes. So this is now the input to the system. They also showed very extensively that this phenomena requires exocytosis of glutamate from astrocytes. And then blocking astrocytes prevented TLTD, STDP in this specific synapse. So these were the guidelines for our model building. And then we used many other literature in addition. So we ended up building this very complex model for astrocyte neural interactions in TLTD induction in this specific synapse. And now I point out it's only this synapse. It works differently in other synapses. So let me walk through briefly. Here we have a presynaptic neuron. We model it as a one compartment. So then we have a postsynaptic neuron, 2 compartments, and an astrocyte process, 1 compartment. We have the endocannabinoid release. It activates calcium, and there are many other calcium sources modeled in this astrocyte. Release of glutamate happens. It binds to NMDA receptor, activates calcineurin, and this is actually the hypothesis, the black box that we are modeling in this work in great detail. So this is the part that we developed. Everything else is very conventional that is used in the current literature. Then this calcineurin induces glutamate release from the synapse and activates these different pathways in the postsynaptic neuron. So this is a very complex model, 80 differential equations, 170 parameters, and the data to construct the model comes from 70 references. So it's not only the Min and Nevian, which kind of provided us the hypothesis, the model, but then we found out the model components from 70 references. And what we are able to show with this model is that we confirm the experimental result of Min and Nevian, and also Banerjee et al. From Paulsen lab. So we saw that the shorter temporal difference, yesterday you learned that you have this difference between pre- and postsynaptic activity to induce STDP. So the shorter this temporal differences, here it's delta t is 10, so you have stronger TLTD. And this is through astrocyte-mediated cellular and subcellular mechanisms, as was kind of indirectly indicated by Min and Nevian. What is the value of this kind of model is that we can now predict the dynamics of molecular reactions. We can, for example, follow the overall calcium during this STDP induction in the astrocyte. We can follow, for example, the calcineurin concentration in the presynaptic terminal, and so on. So this is the value of the model. We can follow simultaneously the dynamics of different molecular entities, which is not possible in the experiments. And we kind of conclude the work that during this STDP, a single astrocyte acts as a delay factor to fast neuronal activity. So many times you hear in the lectures that it's not possible that astrocytes take part in the very fast neuronal processing, and this is completely wrong. During this work, I gradually started seeing that yes, it is actually the key to understand the brain, and this model at least nicely to me, it shows how you kind of integrate the slow activity to this very fast activity to tune synaptic transmission and to induce plasticity and to learn, at least during development. I don't know about adulthood. And we conclude that this is one of the mechanisms that leads to synapse pruning and sensory learning during development. So maybe in the interest of time, we will skip this reproducibility, but you will find the model in the GitHub and Model DP and can look at the appendixes to explain in detail the model. So then I have 2 other examples, but it's only 1 or 2 slides, so we will go fast. So other example is about neuron-astrocyte interactions in networks, because now we have also an interest to understand, like not only in synapse, but at the network level, what happens. there are some tools. Neuron can be combined with Astro. It's very recent development. It's a bit challenging, but it works if you know what you are doing. You can do single cell modeling with Neuron and Astro. There are Brian scripts where you can do calcium dynamics modeling in single astrocytes and networks. There are other tools like steps where single astrocyte modeling can be done, but most of the work so far is done by own code, which is not available anywhere. And then now we are extending NEST to have like phenomenological modeling of astrocyte calcium dynamics, and this is under construction. It's work together with the Jülich people in Jülich, and this is more like tool development. And we would like to model these kind of systems that were introduced to you by Dirk Schubert yesterday. So we are measuring the network activity in cell cultures and doing this kind of workflow for combining experiments and models. And there we need more sophisticated tools that are very efficiently running large numbers of cells. And of course there is later on interest to understand the in vivo networks, as well as was explained by Sasavan Alpada yesterday. So in addition to network studies, we also, within the HPP, we've been trying to understand how neuron-astrocyte interactions contribute to brain signals. And this is a collaboration with, sorry, I'm going wrong way, collaboration with Alain Destex and Federico Tesler in Paris. And there we are interested in modeling the neuronal networks using mean field approach, and then having relatively biophysical models for astrocytes and vasculature and dilation in order to understand the source of bold fMRI signal. So this was the brief tour through a couple of examples what is work ongoing in synapses, networks and brain signals to understand like how astrocytes contribute to the phenomena at those levels. In this presentation, like from the astrocyte cell biology, I would like you to take home this message. Two messages actually. Astrocyte calcium signals vary a lot and depend on the brain area, cell type and developmental stage. And then somatic calcium signals differ from the calcium signals observed in fine astrocyte processes. And many times it's considered that since the somatic calcium signals are slow, because you kind of multiply many sources of your calcium to this signal that you observe in soma, so it looks like that it's seconds. But in these processes you can visualize very fast events as well. Typically neuronal events are in the order of milliseconds. Here we are talking about in processes from 10 milliseconds up to seconds. So it's still different, so it's slower, but it's fast compared to those early in vitro studies that many of us know. Then the second take-home message is about astrocyte modeling, and there we could conclude that modeling astrocyte functions is in its very early phase compared to modeling neuronal functions. And modeling this calcium dynamics requires description of many complex intracellular mechanisms. We need to do this biophysical modeling first before we go to these simplified, more kind of mean field representations of astrocytes. For sure that will happen, like it's happening now for neuronal dynamics, but we are not there yet. And then most of the astrocytic models, they are derived from very early generic models and are based on data from other cell types than astrocytes. And now it's time to build astrocyte models the same way as we have built neuronal models based on data from astrocytes. Also, the tools need to be extended to modeling astrocyte functions. And this is mainly due to the fact that we have recently realized that there is this really big complexity of astrocyte morphology, and also because there is so-called no ground truth measures as action potential for the neuron membrane. So it's more complex for astrocytes. Our ground truth is this calcium signaling, which is really messy. So with this, I think I will conclude the presentation, and then we can discuss more. But I point out to this literature. So there is a big book on neuroglia, and then a computational clear science book where we also contributed a chapter. And this is the people I would like to thank.
Thank you very much. So do we have any questions?
So
is there a question? Yeah? Okay.
Thank you. When autobiographical memories get encoded in hippocampus, somehow these are the examples of one-shot learning. So I would like to know how LTD and LTP interacts in one-shot learning when I mean, there is some instantaneous plasticity, and then suddenly those synapses got frozen and we have these autobiographical memories forever, and if there is any astrocyte neuronal interaction for that.
Yeah, so we have started to look at we have so far mostly looked at the somatosensory cortex, but I have studied a little bit the hippocampus. I know that the mechanisms that are involved in LTP and LTD in hippocampus are differently, and also the mechanisms that astrocytes contribute to this phenomena are different. We most likely start modeling that sooner or later, but they are different. But yes, my answer is yes, astrocytes also contributed during development to these two phenomena in hippocampus. But how you kind of switch between this LTD, LTP, and whether astrocytes is really involved in this switching is not fully understood. We have some indication that they are involved, but at least in somatosensory cortex, but like I said, there is this controversy, and you have to understand that papers that show something like this don't easily get published, because 50% of the groups don't show this astrocyte involvement in LTP and LTD, and 50% of the groups see it. So, and we have not solved this dilemma. I think when the new papers come out, it will be solved. There is the problem. It's related, if one of you know, like to SNARE mechanism, like how you vesicularly release glutamate. So there is a big fight there, but I don't have time to go into details. But when this is shown biologically, then everything is clear, and we can start like more steadily study this. It's more acceptable.
So at this point, there is no experimental evidence for astrocyte involvement in this hippocampal?
There is some, but it's only like in one single paper. It's showing like whether it's involved in LTD or in LTB. But like I said, they don't explain how it switches. Yes.
Okay. Thank you.
I think it has to be modeled as well.
And do we have any other questions? Okay.
So there are two questions on Zoom. Maybe I'll just have one from Zoom when we have Dieter, and then another one on Zoom. So the first question from Rafael Lorenz Delake to everyone, I guess to our speaker. How many astrocyte models approximately you think are needed before we are able to model calcium dynamics for all different cell types, brain areas, etc.? And how long does it take to develop one model?
Yes, thank you. Very good question. So, well, let's put it this way, that once we really have a good biophysical model for at least one astrocyte, then it's easy to kind of copy that to another astrocyte model in another brain area, where you just like fine-tune a little bit the properties of specific mechanisms based on the data from that brain area. So I think once we have one good model, then it will be easier. So it won't take like hundreds or thousands of years. So I think it will happen within the 5-10 years that we will have like the kind of biophysical models for astrocytes that we now have for neurons. And then the second question was that how long does it take to develop one model? Well, it depends so much, you know, like.
But say, given that we have your model and say we trust it, so, you know, based on this, do you think it's just a matter of tuning, or is it more like?
Yeah, it's more matter of tuning. So, like for us now to transfer this to hippocampus, once we have all the data digged out, it will take for Tina, myself and Ausra a couple of weeks, let's put it that way. But it will take a year or two to figure out all the data, to read all the literature. So the bottleneck there is that you need a biologist together with you to read the papers. So typically with us it happens that Tina is the mathematician and Ausra is the programmer, and then together they program and I keep reading the literature. Of course I would like to do the programming, but someone has to really read the literature and understand it. So this is how it goes. So
okay, thanks. So I'll pass on to Dieter and then I'll have another from Zoom.
Yeah, I think that's still similar to neural modeling. It's hard to get enough good parameters to constrain the model, and it always points back to we need more data, especially molecular level data and interactions between these substances. My question was, you showed the LTD was dependent, the endocannabinoid signaling pathway was dependent on calcium release from glia. I wanted to just understand a little better what the advantage, I mean, why not just send the endocannabinoid back to the presynaptic terminal and be done with it? So what is the actual computational advantage of this kind of process?
Yeah, you point to the quick question. Yeah, it's a very good point and a crucial question. So we know that it's been shown earlier, like I think 10 years ago, that the endocannabinoid can also directly activate the presynaptic terminal, and we of course considered modeling that as well, but and it might be, who knows, like if this is the mechanism how you do it later. Like I'm all the time pointing out, we are modeling this very narrow time frame during development. And there really, it's not only the Min and Nevian paper, but many other papers that show that astrocytes are crucial in that. So we think that in this way, the astrocytes are recruited in computation, because it's traditionally considered that they are only involved in this kind of homeostatic function. But now it's becoming more and more clear that they are part of this computation. But we have to always remember that they keep moving. So it might be that at some instances, in some condition, when they are needed for certain specific computation, they move closer to the synapse. And then, like, because the constraints there are so narrow, so you are close enough to kind of also see the endocannabinoid that is released from the neuron. And in this way, you take like additional reservoir to kind of help the computation. So we believe that this is some way of kind of like let's say, we have discussed here the artificial intelligence. Why is it so domain specific? I call stupid, but the domain specific. But this allows you flexibility to the system, you know. So you get all kinds of dynamics out of it, and then you kind of increase the dynamical capacity of the system. And like we don't model this movement, at least not yet, but if it happens, so it might increase the capacity that the system can provide for the kind of dynamical behavior, and then state changes, you know, because then when you recruit all these synapses and you build up these state changes in the and then influence behavior, so it may be additional reservoir for brain computation.
I take it like a function of a teaching assistant.
Yeah, maybe. Yeah, maybe it's a good point. Yeah, teaching assistant.
Okay, so I guess that would be the last question probably from Anja Hartevik. Thank you for this great talk. With advancements in both understanding and model building, do you think it would be possible to model astrocytic tumor cells as well as their interactions with normal neurons?
Yes, of course. So I think there are already some groups that are planning to do this, like Volterra group and some other groups, and there you also need to model the really complex signaling pathways like related to cytokines and such, and how they influence calcium signaling. So yes, this is done, and it will be very important future research field. So actually I see more the modeling of astrocytes for the benefit of understanding the disease brain, like Dirk was explaining yesterday, than to understand the brain computation, you know. So it's more kind of preference for me.
Thank you very much.